Good morning, everybody. My name is Brent Peltola. I am the Executive Director of Partners in Research Canada, and I am pleased to be bringing to you this PIR Live event featuring Dr. Thomas Merritt and his lab from Laurentian University in Sudbury, Ontario. If you have any questions during the broadcast, please use the chat function and we will get to those as we move along in the session. We're going to start with some introductions from Dr. Merritt and his lab team. So I'll turn it over to you guys in Sudbury. Thank you.
Hey, good morning. So my name's Thomas Merritt. I am a professor in Canada Research Chair at Laurentian University. And broadly in my group, we look at the genetics of metabolism. So what we're going to do today is talk to two of my grad students and a technologist from my department about different roles and different career paths in science. So I am a professor. I've been a professional geneticist since the late 1990s, and I've been a professor since 2006. When we talk about the different paths that we've taken to get here, one of my students wanted me to quickly point out I spent 18 years as a student. So all of you who are thinking that being a professor is awesome, you are entirely correct. It is a fantastic job, and it is not a short career path. So I left high school at 18, and I was 36 the first day that I was a professor. So I'm going to give you a quick tour of the lab, but first I want to introduce the people that are with me. So Paul, if you would go next.
Hi everyone. So my name is Paul Guérin. I'm a lab technologist here at Laurentian University in the biochemistry department. So basically that means I teach students how to work properly in the laboratory.
Paul, how long, you've been here longer than I have.
I have just finished 15 years at Laurentian University as a lab tech.
As a lab tech, and you were a student here as a master's student and an undergrad.
And undergrad as well.
Okay. So have you spent more time at Laurentian than you have spent away from Laurentian?
At this point, you're correct. It's pretty much 50-50.
Yeah, all right, that's awesome. Okay, Michelle?
So hi, my name is Michelle Ng. I am a second-year chemical sciences master student in Dr. Merritt's lab. Briefly, my project is looking at how we can use exercise as a way to potentially prevent type 2 diabetes, but we're modeling this in fruit flies.
So, and one of the things we'll talk about is why we work on flies. All right, Jenna.
Hi, I'm Jenna J-Puck. I'm a first-year master's student in chemical sciences in Dr. Merritt's lab. For my project, I'm looking at an enzyme called Glutamine Synthetase and seeing different fly lines that have low, intermediate, and high levels of activity of that enzyme, and seeing if there's a correlation between the enzyme's activity and how they tolerate stress.
So, and I should know this because you're my student. Yes. You're your first year as a master's student, yes, and your second year in the lab.
Second year in the lab.
Okay.
Yeah.
Michelle, how long have you been in the lab?
It's all a blur. Third year.
Third year.
Yeah.
Okay. All right. So one of the things that we started with, there was a bit of a hesitation. So in the lab, we're all on a first name basis. So Paul is Paul, Michelle is Michelle, Jenna is Michelle, and I'm Thomas. Sorry, for some reason my students think that's funny. When we go outside of the lab and we're talking to different groups, we have to decide what the title is going to be. And so when I talk to high school groups, I do usually go by Dr. Merritt, but it's not something that we're used to doing in the lab. So there's always this hesitation of, okay, in this setting, are you Dr. Merritt? Are you Thomas? And so that's one of the things that we have to sort of negotiate in the sort of the professional realm of being a scientist is where are you in a title? Brent, so correct me if I'm wrong, but I thought maybe we would start with just a quick shot of where we are in the lab.
Yeah, that'd be fabulous. If you could just move your thumb a bit. There we go. Beautiful.
My mom is taking these photos for me.
Yeah, that's fabulous.
Yeah,
if we can get a little tour of the lab, that'd be fabulous.
Okay. All right. So this is what we call the wet lab, and it's basically sample prep. And I'll show you in a second the business end of the lab, and it's right there. And it by definition looks really empty, and it's empty because this is where we prepare the samples. And so at the end of sample preparation, it gets wiped down, it gets sterilized, it gets cleaned. So there really isn't a whole lot there. There are a set of pipetters, there's some sample racks, but the point of this area is preparing samples to go elsewhere. And so we've got some reagents that are directly behind me. We've got some other reagents here. We have a technician who's looking at me slightly curiously. See? And then if we go over here, these are fly treadmills. So one of the things that we study is the effect of exercise. And so if you're picturing this, these are fruit flies. So this is a fruit fly. And you should be able to see that. They're pretty tiny. Uh-oh. Sorry, I removed them from the rack and I didn't know where they went back. So to get the flies to exercise, we put them on a treadmill, which is this little thing here. And if you picture a hot dog cooker at a Dairy Queen, that is in fact what we do with these. So we put them on the hot dog cooker and the files turn and the flies exercise. So we use that as a way to promote exercise in the fly. So here we prepare the samples. There we can run the flies. We've got reagents around us, and then we go elsewhere. So there's a room that way that has a spectrophotometer that we use to measure enzyme kinetics. So we do a lot of work looking at how the metabolism of the fly changes in response to one thing or another. There's a lab that way, where we can do metabolomics. So we can look at, what, 20,000 different metabolites? 30,000, I'm behind the times. So we can look at potentially 30,000 different metabolites in a sample of 5 flies, 10 flies?
10 to 15.
So 10 to 15 flies. A fly weighs about a 10,000th of a gram, but no, about a 1,000th of a gram. So at about 100th of a gram of flies, we can measure 30,000 different metabolites in that fly. And then we can look at the expression of 15,000 different genes using either an instrument that's next door to the mass spec that we do metabolomics on, or we can do quantitative PCR down the hallway. So by definition, we've spread things around. We raise the flies in a lab down the hallway. We prepare the flies here, and then we analyze the flies elsewhere, and that keeps things from being contaminated. So you don't want to raise the flies in the same room that you're preparing the flies, because you end up getting a mess of contamination. So that's geographically where we're at, and it's spread out across the campus. Brent, you look like you were about to ask me a question.
Yeah, there's a couple of questions I wanted to throw in there, Dr. Merritt. If you could, we agreed it was Dr. Merritt. If you could tell the audience a little bit about what mass spec is and qPCR.
Yeah, and so we can do that. We can do that now, or do you want to go through and introduce the other people? Or I'll give you a quick primer. So
yeah, I just wanted to bring that up. And the one other question that we do have in the chat right now, and I think it's pertinent to where we are, is fruit fly lifespan.
Yeah, so and we were just talking about this in lab meeting yesterday. So we use flies to model different things. And one of the reasons that we do that is because of fruit fly lifespan. So I'm actually going to make Michelle answer this question because she made eye contact. And so that's one of the cardinal rules. If you actually make eye contact, you have to answer the question. So I'm going to shift this. All right, so what's the lifespan of a fly?
Isn't it like 60 to 100 days?
Yeah, but do you keep them that long?
No. So I usually put my flies on a diet for 10 days and then I sacrifice them. But the reason why we study flies is because they have a shorter lifespan as opposed to, say, like a tortoise. So a tortoise.
Or a beaver. Would a beaver have a longer lifespan?
Yes.
Okay.
Yes. So we'd have to wait years and years and years to sacrifice these animals, whereas in fruit flies, we can wait 3 months and that'll be it.
Right. Okay. So what can you tell about diabetes in humans from working on a fruit fly?
What can we tell? Well, the insulin signaling pathway in humans is actually conserved in insects, so we can actually use fruit flies to study. So we're kind of working backwards. So we don't understand how type 2 diabetes progresses in humans, but we can use the fly to understand that.
Okay. So it fundamentally, the idea is that we're answering questions in the system that we can best answer those questions. And so if we want to understand experimentally how insulin resistance works, there's some things we can do in humans, but experimentation is not one of the things. We just don't experiment on people, but we can experiment on flies. And so we can do a lot of work experimenting on the flies, and one of the things is using that treadmill. So when Michelle puts her flies on a high sucrose diet, they become insulin resistant. We in fact create diabetic flies. And what we're looking to see is if we can delay that transition to insulin resistance by using a light exercise regime. And basically the idea is, are active flies less likely to become diabetic, to become insulin resistant? Okay. All right. Let's hang on to the idea that we're going to talk a little bit about metabolomics. We'll talk a little bit about gene expression, but I want to bring Paul into the conversation. So, Paul, you are tasked with making it safe for people to be in the lab. Right? So you're giving them the skill set that they need so that they don't kill themselves or the people around them in the lab.
Sure.
What do you see your role as a technologist in the department?
Well, on a small scale, my role is really to try and get the students prepared to do a fourth-year thesis. Really, that's the way I see it. Is to try and have them have some basic preparation so that when they come and work for a researcher, such as yourself, they're ready to use some of the equipment and you can put them to good use.
So if we're talking to students that are in high school and in primary school, what are the kind of skills that you would like to see them coming out of school before they come into university?
Well, for sure, math skills is probably one of the top. They need to practice math, and that helps with calculations, and you can't avoid it in a biochemistry lab. You're going to be doing calculations to prepare your solutions. That's even before the experiment starts.
So you're going to teach them the hands-on skills, but you need them to come in with the math skills. What about writing? Do the students in your labs write?
Yeah, there are lab reports, formal lab reports that they submit. Generally, you're looking at about 10 lab reports per semester. Full reports such as includes abstract, results, discussion, references. References very important because we don't want plagiarism.
Right. Yeah. So do you have to teach things like just basic, not scientific method? I can't think what the right term is, but you're teaching beyond the hands-on skills.
Yeah. No. I mean, this plagiarism thing is a big notion that's come up lately, and we need to teach that to students to know how to write a report. If you're not using your own original ideas, you got to tell us where the source is.
Yeah, that's awesome. Okay. All right, so is it okay if I spin around to Jenna and make her tell us what she does?
Absolutely. We'd love to hear what Jenna does.
Yeah, so what's your project? And so again, now Jenna's working with fruit flies, so it's the same idea that we're using a model species. But what is it you're looking at, and why is that important?
So just to give some background first, that my project actually makes sense. So glutamine is an amino acid.
So sorry, I'm gonna interrupt a bit. Thomas, can we get the device a little closer to Jenna so that we have a little bit better audio?
Oh, sorry.
That's perfect. Thank you.
Okay, so so glutamine is an amino acid, and so in actively proliferating cells, so cancer cells, when glutamine is in their environment, they're happy and those cells are going to keep proliferating. But as soon as glutamine is removed from that environment, the cells undergo apoptosis. But there's some disconnect in which like the cells are somehow sensing the glutamine in their environment and relaying that to the cell's survival mechanisms, but we don't know exactly what's happening. And so there's been a lot of great work in cell lines done, but what's really cool with my project is that I'm looking at the enzyme glutamine synthetase, so it converts glutamate into glutamine. And so I'm looking to see, I'll first identify fly lines with low, intermediate, and high levels of glutamine synthetase activity, and then see if there's a correlation with how the flies can tolerate different types of environmental stressors. So I'll be doing heat shock, cold shock, and inducing oxidative stress, which causes DNA damage. And so the main goal essentially is to see if we can use flies as a model organism to further investigate this glutamine addiction phenomenon that we see in cell lines. And from there, maybe sometime in the future, we'll be able to selectively kill cancer cells.
So are you giving the flies cancer?
No, I'm not giving the flies cancer. I am just seeing if they have different levels of activity of the enzyme that they're looking at, and then seeing if there's a correlation with how they tolerate different forms of stress.
Yeah, and so why stress? What's the connection? Sorry, the connection to stress.
So glutamine also plays a role in different stress pathways, and so it forms an antioxidant to prevent what's happening with oxidative stress. So if we can see what's going on with the stress, maybe we can also connect it to that later on.
Yeah. Okay. So I'm pushing Jenna to talk a little bit beyond her comfort zone, but we started the project because we know that there's a role for the enzyme that we're studying in these cancer cells. But one of the common features of cancer cells is this high metabolism, but also a high amount of stress. And so we're looking at the enzyme system that we know has a role in some cancers, but we're looking at its biological role in flies, and looking at a bigger picture. How do we understand the biology of this enzyme that we know in humans has aspects for cancer biology? All right, how are we doing on questions, or do you want to go back to mass spec?
So we do have a question about mass spec. So we'll have to throw out this question and then you can go to there. How do you isolate a sample from the fruit flies to do the spec analysis?
Yeah, so my role in this is absolutely crucial. What I do is I look Michelle in the eye and I say, here's the experiment that I need done. Tell me how it goes. And so then I make her do it. So how do you go from fly to mass spec?
Can I show
The fuck is it? Yeah, sorry.
the?
Oh, yeah. Do I know where the tool is that we do this with? No, I do not. We're getting there. Okay.
Okay, so basically we start with whole flies. We put them. So basically, whenever you put flies, say I put my flies on a 10-day diet, I would freeze them in liquid nitrogen right away, put them in, this is called an Effendorf tube, shown here. Basically, you put buffer in, you stick this nifty little drill in, and you basically ground up the flies until there's basically no fly left. And then we put it in a centrifuge to pellet the exoskeleton. And then all you're left with is just the supernatant or the liquid. So the liquid actually contains all the enzymes that you want to analyze.
Yeah. So we take the fly, we homogenize the fly, and honestly, that's the first step in almost all the experiments that we do. So when we're looking at genes that are expressed, flies have about 15,000 genes, but any given cell might have anywhere from 500 to a couple of thousand genes that are expressed. And the first step is to actually get to the RNA that is being expressed. And so we take the flies and we grind them up using a slightly different grinding system, but fundamentally it's the same thing. You start off with a whole fly and you end up with a liquid extract of the fly. So with metabolomics, we do a liquid extract of the fly, and then we precipitate out all the proteins. And so the only thing left in are small molecules, which we run through a liquid chromatography column to separate out over time. And then we bounce them through a mass spec that gives us a mass to charge ratio. And that mass to charge ratio is so incredibly accurate that you can say, based on this ratio, this is glucose, or this is alanine. And it's not that easy, but that's the fundamental idea behind it, because you've got to identify potentially 30,000 different metabolites doing that. We also do, with that same initial prep, so we start off with a fly, we grind it with a Dremel tool, we work on enzyme kinetics. And so a lot of Jenna's work is actually looking at enzymes. So you've got an interesting twitch and a twist. You have a twitch as well, but an interesting twist in that you want to know what's going on specifically in the mitochondria versus the cytosol. So can you walk everybody through your prep?
Yeah, so I'll grab the tool that I have to use because I can't use the Dremel tool for my project. So for glutamine synthetase, there's two forms of the enzyme. There's one that resides in the
Can you
mitochondria.
turn this a bit, Thomas? There we go.
Oh, sorry. So there's one that's in the mitochondria and one form that's in the cytosol. And so these do, they both still convert glutamate into glutamine. But I'm interested also in seeing if there's a difference between the glutamine synthetase 1 and glutamine synthetase 2 activity. So for my sample prep, if I were to use the Dremel tool, like Michelle uses, when I grind up my flies, it ends up bursting the mitochondria, and all the glutamine synthetase, the mitochondrial form, goes out into my total homogenate. So I would use a glass ounce homogenizer. So basically just has this, where's the camera? This container here, and then a pestle like this. And so I'm able to just gently sort of squish the flies. And so with this tool, what's happening is I'm able to grind up the flies, but in a way that's a little bit more delicate so that the organelles aren't bursting. And so with that, I'm able to separate mitochondria and cytosol.
Yeah. So the idea is flies are actually surprisingly similar to humans. And just like humans, their cells have mitochondria where the energy for the cell is produced. And so Jenna is interested in a form of the enzyme that's restricted to the mitochondria and a form of the enzyme that's restricted to the cytosol. And so fundamentally, we're homogenizing the fly to create this extract, but we're going to do it in a slightly different way because we're trying to harvest a slightly different set of things. And in this case, we're trying to harvest a set of enzymes that are restricted just to the mitochondria. Paul, when you're working with students, so you've got to get students ready to come into the lab and do these kind of techniques. And so together we're going to learn how to use a adult homogenizer or a Dremel tool. But what are you seeing as the challenges for the students when they're in the lab? And what's the hardest thing for them to learn when you're trying to teach new students? Is it really the math and the English? What is it?
I mean, well, specifically, I feel like you threw me like 3 questions there.
Okay.
In the lab, if I'm trying to teach them how to use a piece of equipment. The one thing that we're set up here at Laurentian specifically is we have relatively small groups. So
Yeah.
we only have 24 students in the biochemistry lab. So I can definitely go around and help students use the tool properly on almost a one-on-one basis as I'm circulating through the groups.
Yeah.
But it does take time. Students need lab experience to get better. And that's what the labs are trying to give them.
Right.
Students coming from high school don't have a huge range of experience in the lab, from 0 to some other students tell me they spend, they've done several experiments and they know some of the glassware that we work with.
Yeah.
So that definitely helps. But the university level, we're just trying to keep them familiarized with the various tools accessible in the lab.
Yeah. So Michelle, I want you to speak to that. So you came into the lab initially as an undergraduate volunteer. Why did you start working in the lab?
I wanted to be prepared for when I got to 4th year, what I was kind of getting into, and I wanted to know the lab so that I was ready to go by the time I was in 4th year.
Yeah. And so did you come in as a 3rd year? Yeah. Okay. And so you came in, you volunteered, you learned a lot of the techniques, and then you came back your 4th year and did a thesis. Yeah. So, and that's something students should be thinking about. So 30 years ago, when I was at university, my first year at university, I got a job in a lab washing glassware. And I wasn't learning any complicated techniques, but I was getting my hands dirty and I was talking to other scientists about what they were doing and why they wanted to do it and what their career path was. So what I can say to students that are thinking about college, thinking about university, as early as you can, get your hands dirty and find a lab, either volunteer or get a work study position or something like that, where you can get in and become active in the lab. We've got a student, how long has Jesse started as a 2nd year?
I think so.
So I've had students volunteer as early as 1st and 2nd year to get that experience in the lab. And it can be super important if you want to continue on in a career in science, but it can also be super important in determining this is not what you want to do. All right, so science is a really weird career path. Like I said, it took me 18 years between high school and my first day as a prof. And if it's going to be for you, then get in the lab and get your hands dirty. And if you think it's going to be for you and you get your hands dirty and it's not, it's better to figure that out early when you've got time to look at other options and other things to think about.
That's great.
Okay.
Can we jump into a few questions?
Yeah, please.
So a question that's been sitting in the chat for a little while is about whether or not your lab studies generational effects in fruit flies.
Have we done any generational? Yeah, so most of the work that we do, the flies don't make it past 6 days old. So we harvest the fly, or we collect the flies, because flies have a holometabolous life cycle. So they start off as an egg, they hatch as a larvae. It's a little worm-like larvae. It's called a vermiform larvae. They pupate. Actually, you can see at the base of this kind of, the things that are crawling on the side of this are larvae, and then they pupate. It looks a lot like a butterfly, like a chrysalis or a pupa, moth. No, moths are not butterflies. One of the things, just to be clear, we work on flies and we are not entomologists. So every now and then people come in and they're like, hey, we've got a question. It's like, good, go talk to somebody in biology. I don't know anything about that.
Yeah. There's
So,
a,
so to answer that is no, but there are some really cool studies that do that. So most of the stuff that we do is on adult flies, but we don't last more than 6 days.
cool. There's a lot of interest in how you use the flies. So I'm going to try and combine this into a single question. How do you keep the flies on the treadmill? How long can a fly stay on the treadmill before it gets tired? And how do you keep them moving?
So the hardest thing is to get them to hang on to the guardrails. So in all cases in the lab, we are all about safety first. And so it's a 3-point rule, right? That you have to have 3 points of contact at any given time. And so at any given time, there have to be 3 points of contact. None of my students believe me. So Michelle, it's your treadmill. You explain how the treadmill works.
Okay. No, I guess I can turn it. But anyways, so basically, like Thomas showed, the treadmill sits.
Dr. Merritt.
Dr. Merritt. The treadmill sits in a horizontal fashion, so the vials sit in a horizontal fashion as well. So as there's a conveyor belt that moves below, which slowly rotates the vial. So flies always want to move in the opposite way of gravity. So if we rotate the vial, the flies will always continue to walk up the vial. So it's actually been shown that flies will exercise for 15 minutes at a time, and then we let them rest for 5 minutes. I'm not exactly sure what the total time that they can keep moving is.
But doesn't it vary? So talk about that.
It does, yeah. So there's actually flies that are called high motivation lines and low motivation lines. So motivation is just the tendency of the fly to keep moving. So low motivation lines, I'll just take a picture, sorry. So low motivation lines will stop moving long before the high motivation lines will. So just to kind of standardize this, we keep them moving for 15 minutes at a time and then 5 minutes rest, and do that for a total of 2 hours.
So just to point out, you have shaken those to the point where they're like stuck all over the place. So, but that the difference is pretty cool. It's exactly like if you put the 4 of us in a gym, right? And all of us are going to respond differently to exercise, but we're also going to be more or less interested in walking on that treadmill. And so I'm reasonably okay at walking at a treadmill. I'll exercise a little bit. I can tell you because I've worked out with Paul. He's a monster. And so if you made us work out together, Paul would potentially kill all of us because his motivation to exercise is at a different level. And then Michelle and Jenna would be somewhere in between that. So Michelle would be more fit than I am. And then I might be able to take Jenna if we had to run a long distance or something, right? But the fly lines are very similar. And a line of flies is essentially a family of flies that we raise in these vials. And within a vial, they tend to be genetically similar. And between vials, they tend to be a little bit different, like a different family, but they're all the same species of fly. And it ends up that different families have a different motivation for exercise. And so we put them on this treadmill, the vials rotate, the flies walk. And one of the labs that was studying this a couple of years before we did realized that after about 15 minutes, a lot of the flies would just stop and they would just sit and rotate. And they're like, you know what, I don't care. But what we do is every 30 seconds, the fly goes this way, the vial goes this way. And then after 30 seconds, it reverses and it goes that way. And the flies, that little bit of change, it's like having music on when you're on the treadmill. The flies keep walking. And so they will walk for longer if you change the direction of rotation. But after about 15 minutes, a lot of the fly lines are like, yeah, you know what, I'm done. We're just going to hang out here for a bit. And so we give them 5 minutes. And they literally, it's similar to just sort of catching their breath. And then we do it again. So when you do exercise, what's the exercise regime that you use?
So like I said, so if I have a 10-day diet, I usually do 10 days of exercise. So every single day I'll come to the lab from 1 to 3, starting with 15 minutes. I'll exercise them. I'll hit stop on the computer, let them rest for 5 minutes, put them on for another 15 minutes for a total of 2 hours.
So is the multi-day thing important?
Yeah, so in my project, we're looking at short-term and long-term exercise plans.
And so we talked about the lifespan of the fly. It ends up that 5 days of exercise in a fly lifespan is a substantial part of their lifespan. So flies will live 100 days, but they generally live closer to 30 days, 30 to 50 days. And so within that time frame, 5 days of exercise or 10 days of exercise is a substantial amount of exercise.
So there's a couple of questions regarding the source of your flies. So the one question is, how do you get your first flies, and then where do your fruit flies come from? I don't know. Do you continue to generate flies in the lab?
Who wants to say? Jenna?
So for most of us in the lab, we're using lines that are called, it's from the DGRP, so it's the Drosophila melanogaster genetic reference panel. And so these flies were isolated from a natural population and then inbred over several generations. So let's say if these were two different DGRP lines, if this was one and this was two, then within a vial, because they've been inbred over several generations, there's no variation, but between the two lines, there is variation. We have another project in the lab where another student, we got some wild flies from Sudbury. But basically, if we ever need to get more flies for our projects, we call it just blowing up flies. So we'll take 5 males, 5 females, put them in a vial, and
Then what happens?
then we have more flies.
They make flies.
Yes.
Yes, they make lots of flies.
We make lots of flies.
So we breed and raise a lot of our own flies.
So the other question then that goes along with the, I guess, the differentiation between having multiple lines is, what happens when the flies escape?
They don't escape. We're really good at making sure they don't escape.
Are you?
No, well, sort of. So it's a really good question. And a lot of the flies that we work with are actually from here. So we have flies that we've collected from Sudbury, Ontario, we use for different things in the lab. And if some of those get loose, it doesn't matter. They're local flies. A lot of the flies that we work with are genetically modified. And so you don't want genetically modified flies just being released into the environment. And so we take precautions to try to limit the chance for escape. And by and large, that works, and we don't get a lot of flies that escape. In the rare case that we do get flies escaping, the genetically modified flies are pretty compromised. They're not super healthy. And we don't think that they live long enough to contribute to the population, like the local population. But it's an interesting question, and it's something we have to be very careful about. When we dispose of flies, we take flies that we haven't used, we seal the vials, we freeze the vials, and then we dispose of the vials. So we freeze them at -80 for a couple of weeks, and flies are not freeze tolerant. So if you do that, you kill off the flies. But it is something that we're very conscious of in the lab.
Great, thank you. That was the end of the questions that we had, and we are already a few minutes over time. I don't know if there's anything else that you wanted to make sure that the audience was familiar with out of your lab, Dr. Merritt?
I think what I would leave you with is there are a lot of interesting things that we can study with something as simple as a fruit fly. And there are reasons that we can do it. They're quick to breed, they're small, they're easy to work with, they're experimentally manageable, but we can address things like cancer biology, the biology of insulin resistance. The Treadmill actually was developed for a project where we're looking at safety of miners, so deep underground miners, and how does mining 2 kilometers underground affect physiology. So there are a lot of questions that you can answer with this really simple system. And then the other thing I would leave people with is Paul's comments about the kind of things that you should be thinking about, the kind of techniques, tools, skills that you need for university. And we think about lab skills, and that's absolutely necessary. But fundamentally, I mean, the first thing that Paul said was you need math skills. And I would say that you also need English skills, and we did talk about writing. But if you're thinking about a career in science, you really need to make sure that you have that strong foundation in fundamental math, fundamental English and communication. And that's super important before you go on to university.
One other quick question that I know you're going to want to answer, Dr. Merritt. One of the classrooms is curious how you differentiate the males and the females.
We tickle them. It's not true. So the camera on this iPad is not quite good enough, but if you watch flies crawling on bananas in your fruit bowl, you can actually tell the difference between males and females. So my 9-year-old daughter is really good at determining male and female flies. So the females are a little bit bigger, they're a little bit paler, the males are a little bit smaller, and flies have a chest, a thorax, so the thorax is called the chest, or the chest is called the thorax. They've got a head, and then they've got an abdomen that sticks out the back. And in the females, the abdomens are full of eggs, and so they tend to be a little bit larger and they're pale. So in the males, the abdomen is smaller and it's black. And so you look for the small black flies, and those are the males, and the larger tan flies are the females. So we do it under a microscope and you can see the differences really easily, but even just with the naked eye, looking at the flies crawling around your fruit bowl or your glass of wine for the teachers, maybe 2 glasses of wine, you can tell the males from the females.
Fabulous. Thank you so much. As I said, we are over time, so I'm going to have to cut things off here. I want to thank everybody in the lab. You guys are doing fabulous work. We really appreciate your time talking to our classrooms across the country. To the classrooms that are still with us, thank you for joining us. The recording will be posted in the next 24 hours. And if you're interested, next week, we have another PIR live event, this one featuring Dr. Brian Dixon from the University of Waterloo, who's going to be talking about the aquaculture industry in Canada. So take a look on our website if you want to register for that one. And again, thanks to the lab.
Thanks, everybody. Have a great day.
You too.